Thank you, sir.
Okay, so good evening, students. I am Srijan Sen from IIT Kharagpur. So today I will be taking your week 2 live session of the NPTEL course Experimental Biochemistry, which is being taken by Professor Shomode and Professor Shagota Dasgupta of IIT Kharagpur. So today's lecture will comprise of the amino acids and basically the peptide bonds and all. So you have, I guess you people have gone through the lecture already of week 2 and have also done the assignments. Okay, so I would be also discussing this lecture in brief and also discussing with you a few question and answers that will help you in solving the assignments if you haven't, or if you have any questions regarding the assignments and in the exam which will be upcoming in the month of April and all. Okay, so I'm sharing my slides with you all. I guess it's visible to you all. Am I visible to you all? Just confirm me once so that I can just start. Yeah, great. Okay, fantastic. So, okay, let's start. So today's lecture will mainly comprise of these topics, which includes amino acids and their properties, the isoelectric point, electrophoresis, the isoelectric point determination of glycine, and the isoelectric point, point determination of charged amino acids. Like you have the negatively charged and the positively charged amino acids. So these will be the concepts that will be covered in today's class. Basically, this is a 2-hour lecture, so I won't be able to put focus on each and every point, maybe which has been shown to you or have been taught to you in the entire week. So I would be focusing on the main concepts that will help you in solving the assignments basically, and also there are a very critical portions on these lectures which I would be focusing on, that will help you in also from where basically the questions will come in the exam if you're giving it. Okay, so let's start with the structure of amino acid. So we all know that amino acid means any molecule that has an N-terminal amino group and the C-terminal carboxylic group. So it is attached to the amino. So we have a carbon atom with 4 hands. Our group has so in one hand we have the amino group, in the other hand we have the carboxylic group. The other two hand includes your hydrogen and an R group. So this R group can be anything. Depending on the R group, we can actually distinguish the different types of amino acids. This R group can be depends whether the amino acid is polar or non-polar amino acid, or whether there is a charge involved in this side chain. So on the basis of that, you can distinguish these amino acids into different categories. So what are the different categories? I would be going in details on that. But as of now, if you see from the example that when this particular R group is a hydrogen, that means it is the smallest amino acid or a glycine molecule. Or when this R group is a methyl group, that means it's an alanine. Okay, so this R group, so the moment we vary this R group, the number of times we vary this R group, the different types of amino acids can be generated. Okay, so so what are amino acids? So these are the monomer units which are formed within the long polypeptide chains of protein. So L-alpha amino acids and their derivatives are basically involved in participating in the different cellular functions that are involved in the nerve transmission and biosynthesis of purines, pyrimidines, porphyrins, and urea. So short polymers of amino acids, which we call as the peptides, they have different roles in neuroendocrine system, such as the hormone releasing factors, the modulators, the transmitters, etc. Now, although we know that there can be so the number of times we are varying this R group, there can be as many number of amino acids that can be generated. So there can be even more than 100 amino acids, but only 20 of these amino acids are needed to make all the proteins in a human body and most of the other forms of life. So these 20 amino acids includes the L-alpha amino acids. Okay, so these are basically, so these 20 amino acids are the naturally occurring amino acids which are required to make the proteins in a human body are the L-alpha amino acids. But there can also be the D-alpha amino acids that have different roles in, if you know, the bacterial cell wall, uh, development of the bacterial cell wall, we have a D-alanine over right over there. Okay, so all of these 20 amino acids except for glycine that has where the R group is a hydrogen contains a chiral alpha carbon. So what is an alpha carbon? The chiral carbon. So a chiral atom is one that has four different groups in the hands of carbon. Okay, so the four different groups that combines that are involved in the four hands of a carbon forms an chiral carbon. So all of these amino acids are the L isomers with an R absolute configuration except for glycine that has no chiral center, and cysteine where an S absolute configuration because of the sulfur containing R group. So that's an S and R. So this is about the stoichiometry of the amino acids. Now, humans and other animals cannot synthesize 10 of the L alpha amino acids that are present in the proteins in amounts adequate to support the infant growth and to maintain the adult health. Okay. So, however, you will see that there are certain microorganisms that secretes free D alpha, D amino acids or peptides that contains D and L alpha amino acids. Like they have several of these bacterial peptides are of as well as of therapeutic value, including your antibiotics like the bacitracin and the gramicidin A and the anti-tumor agent bleomycin. So these have contain both D and L alpha amino acids. So certain other microbial peptides are, however, toxic. So when you change a D alpha or you change the orientation of an amino acid, that can add to the toxicity inside the cells that can lead to the cellular growth. And those peptides might not be very much useful for us as they are not involved or they do not have any major therapeutic value. Rather, they add to the toxicity of this. So some proteins contain additional amino acid that arises from the post-translational modifications that already exists inside a peptide. So examples includes the conversion of a peptidyl proline and a peptidyl lysine to 4-hydroxyproline and 4-hydroxylysine, leading to the formation of two other amino acids which are not directly related not directly related to proline or lysine, but they have a different side chain due to the post translational modification, giving rise to two other amino acids. So these post-translational modifications leading to different kinds of amino acids can be due to your methylation, formylation, prenylation, acetylation, so on and so forth. Now, these modifications can or may not have a biological value, but they can add to certain amount of increase in solubility, stability, catalytic ability, and the interaction with the other proteins. Like for in a natural state, for example, a lysine or a protein may be involved in a certain kind of a protein-protein interaction, but this lysine and proline do not show a very high solubility or a stability. But when you are changing them, or naturally what happens when this proline or a lysine is converted to a 4-hydroxyproline or a 4-hydroxylysine or 5-hydroxylysine, what happens, the stability increases and maybe its catalytic activity does also increase. So these post-translational modifications may or may not be biologically very useful, but it depends in which situation or circumstances this modification occurs. If it's occurring naturally or we are doing it recombinantly, so maybe it will or may not have be better for our proteins. So now so now as I was speaking, this changing this R group can lead to the formation or diversification of different amino acids. So here we come. So with the different uh, s- with the different functional groups, as I would say, positioned in the place of R, we can so there are 20 different amino acids that are naturally occurring. There can be many, there can be L and R configuration, but here we are only focusing about the 20 naturally occurring acids over here, starting from alanine, where we have a methyl group, we have a bigger group of arginine, we have asparagine, we have the two acids, aspartic acid and the glutamic acid, which when the COH group is changed with an NH2 group in the when the OH group, sorry, when the OH group is changed with an NH2 group, we give rise to the asparagine or glutamine. Then we have the simplest of the glycine with the complex structures, like we have a tryptophan, tyrosine, phenylalanine. Then we have an amino acid, which is your proline. So there can be many and many amino acids depending on what functional groups we are adding to the R position. Now, it is very important to remember the structure of all of these amino acids, because there can be questions that are related to the structure of an amino acid, to the chirality of an amino acid, and also there can be questions where you might be asked about whether now the structure is very important, the chirality is also very important, and maybe you are given the name of an amino acid and you are asked what is the structure of a particular amino acid, and you are given four options, and from there you have to pick up one. That is why remembering the structure of these amino acids are very, very important. Now, from the basis of here, you can tell whether this amino acid is acidic in nature or basic in nature, whether this amino acid is an aliphatic one or an aromatic one, and whether it is an essential or a non-essential amino acid. Okay, so there are different color codes and different spheres and circles, the dotted circles. So these, there are different ways in which it has been presented. So it looks very good, okay, actually. So the red ones, the red circles indicate that it's an aliphatic amino acid. The green ones indicate it's aromatic, whereas the orange ones are acidic in nature, and the sky blue ones are, the light blue ones are basic in nature. Whereas you have the pink ones that have an hydroxylic group or the OH group, where you have the threonine and the serines mainly. You have the sulfur containing group where you have methionine and cysteine. The amidic groups include your asparagine and glutamine. Now the dotted circles are the essential amino acids, where you have isoleucine, leucine, valine, phenylalanine, tryptophan, lysine, threonines, and the methionine, where the other ones are the non-essential amino acids over here. So questions on the uh, acidity, basicity of an amino acid, the aliphaticity of an amino acid can also be asked. That is why you need to know the structure, the chirality, these informations about all the 20, at least these 20 naturally occurring amino acids. So basically I have two questions maybe for you uh, regarding this. So a pair of amino acids contains non-polar and aliphatic side chains. So you have to identify a pair of amino acids that contains a non-polar and aliphatic side chains. So first, you people give me an answer. I guess you people would be able to answer it. If not, then I would be showing you with the structure. So here you have to identify a pair of amino acid that contains non-polar and aliphatic side chains. I've got one answer. Others also please answer. Non-polar and aliphatic. I would just show you this one once more if you want to just check with the structures. If you're not aware of the structures, just have a glance at this once and probably you would be able to answer it. The question is aliphatic and non-polar. Okay, so what is given over here? Phenylalanine and tyrosine, alanine, leucine, lysine and arginine, that is there. You have serine and threonine. B. Yeah. So let's see. So aliphatic. Let's first identify the aliphatic ones. So the aliphatic ones are your alanine, glycine, isoleucine, leucine, proline, and valine. The others are not. So first you have to identify that. So you have over here alanine, leucine, none of the others match. So basically your option B is correct. Okay. Anyways, so fine. Yes, alanine and leucine are the correct one. Okay, which of the following is an amino acid? Okay, proline I just mentioned about it. It's an amino acid. So proline is the correct option. I'm not going into the details. Okay, now about the chirality of the amino acids. Let us try to answer them. So the number of chiral centers in isoleucine and leucine. Okay, so you have to find out the number of chiral centers which isoleucine and leucine have. I would just show you the structures of them. So you have isoleucine and leucine. So you have to find out the number of chiral structures in isoleucine and leucine. And what are the options over here? So it is 1 and 2, 2 and 1, 2 and 0, and both are achiral. So isoleucine and leucine. So I've got an answer of A, 1 and 2. Okay, let's see what others tell. Okay, 2 and 1. So you're telling B. Okay. So chirality is the if the carbon has 4 different groups in its 4 different hands, it means the carbon is chiral one. Okay, so if it has 4 different functional groups, it is a chiral one. As simple as that. So now tell me. So if we look for isoleucine, what happens? The first, this carbon or the alpha carbon is basically chiral because we have NH2, this group, and we have COH, and over here we have H. What about this carbon? This carbon has a CH3 group. You have a CH2 CH3 group over here. You have a hydrogen over here and this bigger group over here. So even this carbon is a chiral one. This carbon is not chiral because over here you have two hydrogens. So this is a chiral. Now, so over here you have 1 and 2. Now for leucine, this one is automatically a chiral one, a chiral center. But for the other two, here you have two hydrogens and over here you have CH3 and CH3. So both of them are basically a chiral. So that is why the correct answer over here is 2 and 1. So those who have thought of it, 2 and 1, that is absolutely correct. Now, next one. Tyrosine and tryptophan. Tyrosine and tryptophan are less hydrophobic than phenylalanine. Why is it so? So why is tyrosine and tryptophan less hydrophobic than phenylalanine? So what options you have? Phenylalanine side chain has a phenol group in it. Phenylalanine side chain has no polar groups attached to it. Tyrosine and tryptophan have similar side chains. Phenylalanine side chain has indole group attached to it. Okay. So which of this is correct? Tyrosine and tryptophan are less hydrophobic than phenylalanine. Yes, absolutely. Phenylalanine side chain has no polar groups attached to it. That is why. Okay. Now, so here we will speak about the 21st amino acid. So humans contain approximately 2 dozens of selenoproteins that includes certain peroxidases and reductases. So selenoprotein P, which circulates in the plasma. Now peptidyl selenocysteines is not the product of a post-translational modification, but rather it is directly inserted into a growing polypeptide during translation. However, like the other 20 amino acids, incorporation of selenocysteine is specified by a large and a complex genetic material for the unusual tRNA called the tRNA or tRNA SEC. Okay, so it codes for maybe a stop codon. So if the tRNA is designed such that if it identifies a sequence of the stop codon, it does not stop over there, because there is no tRNA which is designed for a stop codon, or a stop codon does not have a tRNA. So if we design a selenocysteine where we can incorporate this particular 21st amino acid into the place of a stop codon or in an unusual position, then it is basically possible. So the classification of amino acids can be done in two ways, like you have the non-polar hydrophobic ones and the polar hydrophilic ones. So the non-polar includes your aliphatic and aromatic, whereas your polar amino acids are acidic, neutral, or basic in nature. So all 20 amino acids differ in the chemical structure, volume, and shape. At room temperature, all of these 20 amino acids are crystalline solids. They are colorless, water soluble, and at neutral pH they exist in a zwitterionic form. So let us identify the polar amino acids over here. So polar amino acids includes your acidic, neutral, and the basic amino acids. So the acidic amino acids are your aspartic acid and the glutamic acid, The basic amino acids are arginine, lysine, and histidine, whereas your neutral amino acids include cysteine, asparagine, glutamine, serine, and threonine. So the polarity of the amino acids depends on the side chains. So the polar amino acids make hydrogen bonds with the suitable groups, like you have the OH group or the NH2 group that interacts with the water molecules, making them more soluble. And the polarity of the amino acids actually affects the structure and the function of the protein that leads to some sickle cell anemia, a severe kind of a disease. Now why does that happen? Because we know that proteins molecules, now peptides are the building blocks, or the amino acids are the building blocks of a protein. We always cannot understand from the perspective of a peptide, because we can only understand it when these peptides together form a amino acid. It forms a protein. Now inside a protein, how does it affect? We can only understand on the basis on how these amino acids are arranged. Now the arrangement of the amino acids are very, very important in determining the structure and the function of a protein, because if you have a hydrophobic molecule or a hydrophobic peptide that is to the outside of a protein, what it would try to do, it would try to run away from the environment that is full of water molecules. So the hydrophobic, we call it the hydrophobic core of the proteins, where only we have the hydrophobic amino acids inside it, and the water soluble amino acids are outside it interacting with water, which determines the structure of the proteins. That is why it affects the overall structure and the function. So the hydrophilic and the hydrophobic amino acids are distinguished, and they have been put in the two different columns. So now the essential amino acids. So I was speaking, when I was showing you the structures, I was speaking about essential amino acids and non-essential amino acids. So humans can synthesize about half of the amino acids needed to make proteins. The other amino acids are called the essential amino acids that must be provided in the diet. Those are not required to be provided in the diet. Those are the non-essential amino acids. The 10 essential amino acids includes your arginine, valine, methionine, leucine, threonine, phenylalanine, histidine, isoleucine, leucine, and the tryptophan. So proteins that provide all the essential amino acids in about the right proportions for human nutrition are called complete proteins. Now examples of complete proteins can be Now, examples of these complete proteins are those that you can get in meat, fish, and milk and eggs. Now, about 50 gram of complete proteins per day is adequate for the adult humans. Now, proteins that are severely deficient of one or more essential amino acids are called the incomplete proteins. Now, if the protein in a person's diet comes directly from one incomplete source, the amount of human protein that can be synthesized gets limited to by the amounts of deficient amino acids that can lead to different kinds of diseases and disorders. So you can also get a few rare and unusual amino acids that are generated by the post-translational modifications. I was speaking about it. So you have a different form of proline and lysine over here. You have the 4-hydroxyproline and the 5-hydroxylysine. So in addition to the standard amino acids, the other amino acids are found in proteins in smaller quantities. So these are called the rare amino acids, even though they are commonly found in the collagen. Now, some of the less common de-enantiomers of amino acids are also found in nature that are generated by different kinds of bacteria and all. Some can be toxic, some can be beneficial for a bacteria, like you have the D-glutamic acid that is found in the cell walls of many bacteria, and D-serine, which is found in many earthworms. So some naturally occurring amino acids are also not alpha amino acids, because as I was speaking, this has to be, so the proteins are mainly composed of the L alpha amino acids. So some may not be alpha amino acids, like you have the gamma aminobutyric acid or GABA in the neurotransmitters in the brain. You must have heard of it. And the beta alanine is also a constituent of the vitamin pantothenic acid. Okay, so these are the rare or the unusual amino acids that also do exist. Now we need to understand the properties of the amino acids from an acid-base perspective. Now, although we commonly write an amino acid like we have the NH group followed by C alpha carbon attached to an R group and an H group, and then we have the carboxylic group. But in a neutral pH or inside our body, how do we have them? Or even if we have them, how do they exist? So they exist in a dipolar ion form. So this dipolar ion form, what does it mean? That when we are writing it as an amino group, so the amino group or the NH2 group exists as an NH3 plus, and the COOH group exists as COO minus. So we have a positive charge in the N terminus of the amino side, and we have a negative charge in the C terminus or the carboxylic side. So this dipolar ionic form is also known as the zwitterionic form of an amino acid, which is important for understanding the acid-base property of an amino acid. Why I would be telling, I would be coming to this in my later slides. Okay, so the dipolar form of these amino acids gives some unusual properties. Like you have the amino acids which have really high melting points, like over 200 degrees Celsius. So like for glycine, which has a melting point of 262 degrees Celsius. Now, amino acids are more soluble in water than they are in the organic solvents like ether, dichloromethane, and so on. Now, amino acids have much larger dipole moments than the simple amines or the simple acids. You can see that from the dipole moment value of glycine. Then you have for it's 14D, but for propylamine it's 1.4D, and for propanoic acid it's 1.7. Okay, so most amino acids are also less acidic than carboxylic acids and less basic than most amines. So in fact, the acidic part of the amino molecule is the NH3 plus group and not the COOH group, and the basic part is the COO minus group and not the NH2 group. And it is a very important point for you all to remember. Now, because these amino acids contain both an acidic and a basic group, we can call them as an amphoteric in nature. Now, their predominant form of the amino acid depends on the pH of the solution, in what condition they are in. In an acidic solution, the groups are protonated to a free group and the molecule has an overall positive charge. When you raise the pH to a basic level, it loses a proton, and this is the first dissociation constant. And when it is further raised, if it has exchangeable hydrogens, it will exchange it, and that's the second acid dissociation constant. If you have more that have exchangeable hydrogen attached to it, then it will have more than 2 then 3 acid dissociation constant. Now, above a certain pH value, the overall charge of the protein becomes or overall charge of the amino acid is negative. Like if you see the example, when we are considering it only for like we have only R and it has only one you have two replaceable hydrogens right over here. So you have the pK1 and the pK2. So we have the NH3 group, then we have the COH group. So first the hydrogen from the COOH group is lost. That is your first acid dissociation constant, and then the hydrogen is lost from the NH3 plus group, and that makes the amino acid anionic in nature. So previously it was cationic, positively charged, and then after losing the two hydrogens it becomes anionic in nature. Now isoelectric point is the one, so it is, you can call it in that pH where the, how do I explain it? So an amino acid has a positive and a negative charge, whether it is in the acidic solution or a basic solution. So there must be an intermediate pH where the amino acid remains balanced. Okay, like you have the neutral pH over here. Okay, so the pH or the range of pH in which the amino acid exists in the neutral form, we call it the isoelectric pH or the isoelectric point, pi. So it is cationic, here it is anionic in nature, but at the middle pH where it is in neutral, it exists as a zwitterion. We call this particular pH as the isoelectric pH. Now, side chains of aspartic acid and glutamic acid contains acidic carboxylic groups. So these amino acids have acidic isoelectric points around 3 to 4, and then acidic solution would be needed to prevent the deprotonation of the second carboxylic acid group and to keep the amino acid in its neutral isoelectric state. Whether for now, however, for a basic amino acid like histidine, arginine, and lysine, the isoelectric point is moreover to a basic value because the second hydrogen that will be lost from the NH3 plus group will be towards a higher pH value, and a basic solution would be needed to prevent the protonation of the basic site. So the other amino acids are moreover considered neutral with no strongly acidic or basic side chain. So their isoelectric points are slightly acidic from 5 to 6 because of the NH3 group is slightly more acidic than the COH minus group that is more basic. Now, to understand the acidity and the basicity of these amino acids, the process of electrophoresis is very, very important. So, electrophoresis is a procedure that uses the differences in isoelectric points to separate the mixtures of amino acids. A streak of amino acid mixture can be placed at the center of an acrylamide gel and or a piece of filter paper in a buffer solution, and on the two ends you attach you pass current through the two ends, you attach across the two electrodes. The positively charged amino acids would be attracted towards the cathode, and the negatively charged amino acids would be attracted towards the positive electrode or the anode. An amino acid that will have no net charge will show no mobility. Now, this is an experiment which most of you can do it, or most of you have probably done it in your lab. Like you have a mixture of alanine, lysine, and aspartic acid in a buffer solution of 6. Now, if we look into when we know about the structure of the amino acids or the properties of these amino acids, it would be very easy to say like alanine is something, lysine is something, and aspartic acid is something. Okay, but when this experiment was done, probably people never knew what these amino acids are, or the structure of these amino acids were not basically known. So now you have an alanine, lysine, and an aspartic acid in the center of this gel cassette, and you are passing power over here. So you have the cathode over here and you have the anode over here. So what do you understand from here? Like the anions or the negatively charged thing will be moving towards the anode, and you have the cations moving towards, or the positively charged moving towards the negatively charged electrode, or the cathode, like, right. So as we know from the already know from the structures, like for the isoelectric point for a pH for a lysine, the isoelectric pH for lysine is about 9.7. For aspartic acid it's about 3, and for alanine it's about 5.8 to 6. So when a buffer solution of pH 6 is already present over here, so alanine being neutral in that particular buffer condition will not show any mobility, and that what happens. So alanine does not move. For aspartic acid at a higher pH than its isoelectric point, like when the pi is lower than your original pH, or the pH is higher than your pi value, then it is negatively charged, and when the pH is lower than your pi value, it is positively charged. Therefore, lysine will be positively charged, having a isoelectric pH of 9.7, whereas aspartic acid will be negatively charged, um, depending, like it has a pH of isoelectric pi of 3. Okay, so lysine moves towards the cathode, aspartic acid moves towards the anode, alanine does not move. Now after a period of time, these separated amino acids can be recovered. You can scrape out the bands and use a reagent of ninhydrin to make the bands visible, and you can identify these amino acids by comparing with the known standards. So this is a very easy experiment which you can do, and this will also tell you about the charge of the amino acids. But what is important over here is that the 2 points that I mentioned, when pH is equal to pi, it is neutral and it does not move, like if it's for electrophoresis. Otherwise it's neutral. When the pH is greater than pi, it is negatively charged, and when the pH is less than pi, it is positively charged. Okay, so from here we can understand the titration curve of glycine. So what happens over here, the curve starts at the bottom left, where the glycine is entirely cationic in nature. So there is a pH of say probably 0. So the base amount of base like NaOH is slowly added drop by drop, and the pH is recorded. At pH 2.3, what happens, half of the cationic form has been converted to the zwitterionic form. Okay, so over here what happens at this particular pH, so half of them are in zwitterionic form and half of them are in cationic form. At pH 6, all the glycine molecules are in the zwitterionic form. So at this particular point, or at the base like pH 0, you have the cationic form of the glycine molecules over here. Over at this end point, or at pH set 12 or more than that, you have the anionic form of glycine. In the middle point is the zwitterionic form. But pKa, or pKa1 and pKa2, are basically the midpoints of these two. So you have 0 plus 6 divided by 2, something, some value you have over here, and may say like the pK is 2.3. So at 2.3, you will have half of the cationic form and half of the zwitterionic form. Similarly, at pK 9.6 or at pH 9.6, what you would be having is half of the zwitterionic forms would be converted to an anionic form. So 50% anionic form and 50% will be zwitterionic form. When it reaches a pH of 12, you will have the entire of the glycine molecules converted to an anionic form. Okay, so how do you calculate the isoelectric point? So pi is basically pKa1 plus pKa2 divided by 2. Pi is as simple when you do it for the neutral amino acids, but it does change when the amino acid is a charged one. Now let us see what happens when the amino acid is charged. For example, in the first case, you have a negatively charged amino acid or an acidic amino acid. You have the glutamic acid over here. So like glycine, let us try to explain what happens for a glutamate. So at the base point where the pH is close to 0, all the glutamate ions or all the glutamates are positively charged, but you have only one NH3 plus, you have COOH and two carboxylic groups over here. So the total charge at this particular point is +1. Okay, now the you add base drop by drop, you start adding the base. Now what happens over here is there is a point, say at when you have added about one equivalence of your the base. At this particular point, you get the you get a neutral pH, because the hydrogen, the replaceable, the hydrogen on this carboxylic which is attached to the alpha carbon will start to get exchanged, and when it starts exchanging, it forms a zwitterion. Okay, and then at a particular pkr, which is your middle point where you are, or there is an exchange of the hydrogen from the R group. So this is represented as the R group over here. So when the hydrogen is replaceable with the when the hydrogen of the R group is basically replaceable, you mention it as the pKr, and pK2 is when the hydrogen also loses from the site the NH3 plus. Okay, so when this hydrogen is lost, you mention it as the pK2. Now here you have three pKs, or the three equivalent points, pK1, pKr, and pK2. So how do you know which two pKs values to take and calculate the pi? So for this, there are two ways. The first way is when you see that what happens for glycine, you go from +1 to 0 to -1. That is the value that you are taking. The exchange from -1 to +1. The two equivalent points that are involved between -1 and +1 are the should be taken. The average of these 2 points should be taken, because in between these 2 points there is the zwitterionic form, because you have a plus and minus in the zwitterionic form. So similarly, you can consider a same case for glutamate, where you have +1 to the exchange to -1. The two pK values between these two, the average of these two values can be taken, and the middle point can be considered as the isoelectric point. Like here you have 2.2 and 4.2 divided by 2, so about it will come 3.2 something. Okay, now similarly, the same thing, the same kind of a situation happens for your arginine or a lysine or a positively charged amino acid as well. So here you have three pKs, but in this case, like you have +1, 0, -1, and +2. Here you will have +2, +1, 0, and -1. Okay, so first you have the chart. The amino groups will be positively charged. You have NH3 plus on both the amino groups. That is your where the charge on the amino acid is +2 at this particular point. When you add base drop by drop, it start the hydrogen on the carboxylic group starts to lose, and then the charge becomes +1, followed by the amino group that is adjacent to the alpha carbon, attached to the alpha carbon, loses its hydrogen, and that is the zwitterionic form that you get over here, and that will be your pi value. And at -1, or at the end point, the entire of the hydrogens are replaced and the charge on the amino acid is -1. But for also here also, for the calculation of the pi value, what you need to do is you have to see the exchange from +1 to 0 to -1, or vice versa. You can also think -1 to 0 to +1. You take the two equivalent points of these two, you take the average of these 2 points, and you get the pi. Like you have 9 over here and 12.5, so 9 plus 12.5 divided by 2, it is about 10.8. So that is the pi of the amino acid. So these are the ways of how you can do it. That is the most scientific way. The other shortcut way of how you can do it is, you must be, when you look at the amino acids and the pK values of these amino acids, you see that for a acidic or an acidic amino acid, there are two pKs that are very close to each other, and one pK is very far away from the two of them. Whereas for a basic amino acid, you have the two pKs which are very close to one another, and one pK is very far away from over here. Like here you have 2.2, 4.2, and the other one is about 9.7. So what you do, the two pKs that are close to one another, the average of those two should be taken. Like here you have 2, 9 and 12.5. So 9 and 12.5 are very close to one another. So you take the two of the average of these two and you do it. But that's a very shortcut technique. It might not be applicable for all the amino acids. So the other way out is when you do it in this manner. That will also help you, because there might be questions where you can be asked what will be the structure of the amino acid when the amino acid is in the +1 state, or in the zwitterionic form. Therefore, it is very important that you are aware of the structures even at the different pH. And from there, you might be also asked to calculate the PI and the maybe the PK, A1, 2, 3, or the PKR as well. Okay, so this is about the titration curve of the glycine and the acidic and the charged amino acids. So this is mostly about, so this lecture is mostly about your introduction to amino acids, the properties of the amino acids, the structure of the amino acids, and about the isoelectric point of these amino acids. uh, I think there is not much to discuss about this anymore. So I would be now discussing the question and answers. But before that, if you have any questions on the lectures, I would be happy to answer them. And then we can move on to the uh, solving a few practice problems. If you have any problem in any of the slides, just ask me. Questions from these lectures, there will be many questions from these lectures in the exam as well. And some people might be giving jam and CSIR, so there are a lot of questions from here. It's a very scoring portion, so just don't miss these portions, because it's very scoring, as well as once when you can catch these things, you need to clear your concepts as well. And it's very much you can easily get correct answers from these portions if you can understand the logic and all. So no questions or any, I guess people you have understood. If that is the case, then let's start solving the questions, and then maybe if you have any doubts, we can get that clear. Okay, so let's go on to the first one. Okay, so examples of amino acids containing non-polar aliphatic R groups. A similar question like I discussed. So you have to find out the amino acids that contains non-polar aliphatic R groups. Tell me, this is an easy question. Are you guys there? So what are the options over here? You have phenylalanine, tyrosine, and tryptophan. Okay, then so one of them contains a polar group over here, so that is not correct. You have methionine, alanine, and leucine. So all of them three are aliphatic as well as they are non-polar. So that is one option that is correct. You have lysine, arginine, histidine. That is not correct. You have serine, threonine, and cysteine. That is also incorrect. So B will be the correct option over here. If you have doubts, just ask me. And I expect you people to give me an answer because that will help you to also, and I want people to interact. Okay, so just put up an answer. Maybe you're not sure of it, but it's always good to try. Next one, which of the following statements is true? Tryptophan and tyrosine are significantly more polar than phenylalanine. Leucine is commonly used as an ingredient in the buffer of SDS page. Aspartate is an essential amino acid. Lysine is a non-essential amino acid. Which of the following statements is true? Similar question was discussed a few slides back. Am I audible to you all? No answer. Okay, A. Okay, if whether it's an essential or a non-essential amino acid, we can get it from over here. Like you see over here, it's a non-essential amino acid and essential amino acid. So aspartate and lysine. So lysine is an essential amino acid and aspartate is basically a non-essential amino acid. Okay, so let me check. So C and D is incorrect. Leucine is not used in the buffer of SDS page, rather glycine is used. Therefore, A is the correct option because tryptophan and tyrosine are more polar than phenylalanine, and that is true. Next. Okay. Which of the following statements is or are true for isoelectric point? The pH where the molecule carries no net electric charge, pH where carboxylic group is uncharged, the pH of maximum electrolytic mobility, and the pH where all the groups are titrated. Which of the following statements is or are true for isoelectric point? These are easy questions. All of you should be able to answer it. So what happens for an isoelectric point? So it's the zwitterionic form where the amino acid is in a neutral state. So the pH carries no net electric charge. That is true. The pH where the carboxylic group is uncharged, that may or may not be true. The pH of maximum electrophoretic mobility, that is incorrect. Where the pH groups, the pH where all the groups are titrated, that is also incorrect. Okay, so A is only the correct option. The next one, which of the following amino acids are not optically active? Which of these amino acids are not optically active? So what is optical activity? So when a carbon atom has four different functional groups in its four different hands, it's said to be chiral. Now those atom or those amino acids which are chiral are actually optically active. Now which of them is not chiral, or the other way around, which of them is not optically active? So we have only one amino acid that is not chiral as well as not optically active. Identify that. It's basically glycine, right? It's glycine. Fine. The next one. Okay. So let's look into the first one. Which among the following have the most chiral centers? Which among the following have or has the most chiral centers? Isoleucine, serine, tryptophan, and threonine. Isoleucine, serine, tryptophan, and threonine. Most chiral centers. So we have seen before, isoleucine have two. How many does serine have, tryptophan have, and threonine have? Okay, A. It's asking the most chiral centers. The most. Okay, let's go back to the structure once again. Let's try to understand the structure of serine. Where do we have a serine? Okay, so serine. So serine has one. Where is tryptophan? Um, okay, so where you have the chiral center? This is the one. Okay, you don't have any other. This is not chiral. You cannot call this chiral. These things are not chiral. Okay, so even tryptophan has one. What about threonine? Where is threonine? Okay, so one group is this. You have a methyl group over here and an H over here. So one, and the other one is this. So two. Okay, so two for threonine, and isoleucine also has two. So A is not correct. A is not correct because A is showing 2 and 3, right? A is 2 and 3. The answer should be 1 and 4. So it is D, isoleucine and threonine. Okay, the next one. Calculate the pI of an amino acid that has a pK1 of 2.16, pK2 of 9.06, and pKr of 4.56. So what will be the pI of such an amino acid? So simple. Very simple. Pi is equal to pK1 plus what? So the two PIs which are the two pH which are very close to each other. So here I will take pKr, okay, and the average of the two, which is 2.16 plus 0.56. How much is it coming? So B. Okay. Next one. Okay. So the first one, choose the correct option. Which of the following is a set of polar amino acids? You people give me an answer first. Which of the following is or are a set of polar amino acids? Phenylalanine, tryptophan, alanine, lysine, proline, methionine, serine, glutamine, glycine, uh, glutamic acid, asparagine, and threonine. Which set contains polar amino acids? Polar amino acids. Alanine does not contain a polar amino acid. Alanine is not a polar amino acid. So is phenylalanine. Okay, so the first one is not correct. That means A and D cannot be correct. It has to be either 3, 4. So let us first, so 4 is correct because you have the glutamic acid, asparagine, and threonine. So these are all polar in nature. So 4 is correct. What about 3? So you have serine, that is polar, glutamine, that is also polar, but glycine, is glycine polar in nature? It's not. Glycine is not polar in nature. If I'm glycine is an aromatic side chain, it is not polar in nature, so it should be the answer over here should be only 4. Okay, so it is only B. The next one, which of these amino acids have pI close to the physiological pH? So the physiological pH or the pH of the blood is 7.4. So which of these amino acids have a pH that is close to or has a pI that is close to the 7.4? Histidine, serine, asparagine, and glutamine. So the answer is histidine. Now, the next one, the one letter code for the tetrapeptide. So you have a tetrapeptide over here which has phenylalanine, glutamine, tryptophan, and aspartic acid. So the one letter code for this tetrapeptide should be what? You have PGTD, FNTE, FQWD, and FQYE. A. So phenylalanine glutamine, tryptophan, and aspartic acid. Yeah, if you go by the first letters of each of these, what you answered is correct, but that is actually not the right answer. For phenylalanine, it is F. For glutamine, you have Q. For tryptophan, you have, excuse me. So for tryptophan, you have W, and for aspartic acid, it is D. So C is the correct option. It's not by the first letter of each of that. It does not go by that. Each of them have a one letter code. So that is also another question that you need to keep in mind, because there will be a few around, like find the one letter code of these. Okay. Anyways, so the next one. When the amino acid alanine is added to a solution with a pH 8, alanine becomes. So when you have a pH 8 buffer and you add alanine to it, what does it become? Does it become positively charged, negatively charged, partially positively charged, or does bitter ion? So the pI of alanine is basically 6. So at pH 6, so the pI is 6 and you have a pH of 8. So just I was mentioning, like if the pH value is higher than the pI. So if pH is higher than pI, what happens? It becomes negatively charged. Okay, so B is the correct option. The next one, you have an aspartic acid solution to which you are adding NaOH drop by drop, starting from pH 1.5. So the number of inflection points that you would be obtained. So the inflection points means the sharp changes. So how many sharp changes would you be observing when you are adding NaOH solution drop by drop to a solution of aspartic acid? The other way of saying it is that how many equivalent points do you observe? So for aspartic acid, you have, it's a negatively charged so it's an acidic amino acid. So you would be having three equivalent points, pK1, you have the pKr and the pK2. So the changes over here is the first one from 1 to R and the other one is from R to 2. Okay, so that is why you have two inflection points. Two sharp changes would be observed in the graph or the titration curve for aspartic acid or even a basic amino acid as well. Okay. The next question is, during pH metric titration of glycine with NaOH, certain amounts of HCl is added at the beginning. So what is the role of this HCl? Is it to neutralize NaOH, neutralize glycine, completely protonate glycine, or converts glycine to zwitterionic form? So you have to tell me the role of HCl that is being added at the beginning. No? So it is not for neutralizing NaOH, as you have not yet added NaOH, because it's added in the beginning. It's not for neutralizing glycine, rather it is for completely protonating the glycine. And therefore this is very important, because if the glycines are not completely protonated, the middle point between the between the positives of the glycine in the positive form and the zwitterionic form of glycine, so the middle point will shift severely, and this would result in the this would lead to a discrepancy in your results. Okay, so for completely protonating glycine, HCl is added in some amount at the very beginning of the experiment. Okay, so next one, the peak. Yeah, do you have a question? Yeah, if you have a question, you can just ask me. Yeah, sorry, just got. Yeah, so do you have a question? Any question? Right. Okay, so the next one. So the pK2 or the pK for the side chain of histidine is 6 when it is present in isolated state. Considering a specific protein with histidine at its active site, the pK2 is expected to be. So what happens when histidine residue is present in an active state? Um, when a histidine residue is present in an active state that has mostly the positive charges, the pK2 becomes lower than 6 because of the positive charges in its active site will destabilize the positively charged histidine side chain. So higher concentration of protons, that is lower pH, would be required to protonate the histidine side chain, thereby resulting in the lowering of pK. Therefore, less than 6 if the active site has more positive charges would be the correct option over here. Okay. So titration of arginine residue present in a protein is difficult to carry out with NaOH, unlike individual arginines. Why? So we don't have many people because of there was an error in the connection, so most of them have left. So I would just try to give an answer to this quite quickly. Okay, so what happens over here is that in the arginine present in a free state is different from that in a protein structure, because at extreme pH conditions the protein structure might get degraded, but what happens on the other end is that if it's the amino acid in an individual, it only affects that particular amino acid, but inside a protein there are other amino acids as well, so which might get affected. Therefore it leads to the loss of structure, function, and also the data that we might be getting is not at all that very good. So the multiple amino acids may make the process complex, and the structure of the protein might degrade due to the higher pH. So 2 and 3 are the correct options over here. Okay, and okay, so that's all. So we do not have any more questions. So that is all then. Thank you for joining. If you have any questions, you can just drop me an email. Okay, I would be very happy to answer them. If you have questions from this slides, or even if you have it regarding the assignment, I would be very happy to clear your doubts. Okay, so I'm just dropping by my email. If you have, you can just put it. And secondly, it's that this lecture is being recorded, so those who are missing it, maybe you can get it from the YouTube, and the slides will also be shared with you in the uh, by the NPTEL team. Okay, so thank you all for joining. We will again meet in the next class next week, same